This class is going to be divided into two parts. The first part, I'll be solving the remaining 25 questions from our 100 questions. And the other part of the second part, that's where we're going to do a recap on the whole subject. Okay, so I'll start with this question. The question here talks about farming plant crops that's more energy efficient than animal farming. So again, so as if someone's asking you like why consuming animal products is less energy efficient than plant products. Usually with plants, you get like way more energy, get the energy from the source. That's because you're at a lower trophic level. Think about it that way. You have the option to eat 2 meals. You've decided to eat beef. The beef has already lost a lot of energy because you are here at the third trophic level. So if you've been at the second trophic level, you would have had way more energy. Reason, you always lose energy as we go from one trophic level to another. The processes responsible for the loss of energy are those of Mrs. Grant: movement, respiration, excretion. All of these are responsible. Now, usually the rule says that 90% are lost, 10% are passed on. So only 10% are passed from one trophic level to the next one. In addition, many of the animal parts, like think of a chicken, many of the parts of the chicken are not edible, right? So those parts, they're not going to be used. And that mostly, again, applies to animal products, right? Now, lastly, it's the type of farming. Think of a plant farm and think of an animal farm. Which one requires more energy, more electricity, more resources, more money? It's the animal farming. So for all these reasons, it's way better to consume plant products. Question 72. This one talks about the pyramid of biomass and why is it always upright? Why isn't that's not the case of pyramids of number. Pyramids of number might be inverted. So reason, that's because in the pyramid of number, sometimes you get large number of a small insect. Like in my case here, you got a lot of those caterpillars in one tree, right? Also, that means what? That means that the representation of those insects will be a big block. But that's not the case with the pyramid of biomass, because the pyramid of biomass is always upright. What makes the pyramid of biomass always upright? The fact that we're measuring the amount of energy. The mass of a leaf is like a reflection of how much energy there is in that leaf, right? So for that reason, it would always be, producers would always have more energy than the consumers. Okay, so if you weigh a tree, the biomass of the tree or the amount of energy stored in a tree is far more than those stored in a herbivore. Okay. Question 73, which is super important. It's talking about why number of top carnivores are always limited, are always less than the numbers of herbivores. It's like, do you see all these three questions we solved so far? They're all like trying to answer the same question. We're wasting energy because of all these three processes as we go from one trophic level to the next one. So look what I'm going to say here. I'm going to say only a small percentage of light energy is used. So the plant is far from being efficient. Photosynthesis itself wastes a lot of energy because light energy during photosynthesis, or before photosynthesis, is reflected. Sometimes it penetrates and it doesn't hit the chlorophyll. So for all these reasons, the energy is wasted even before photosynthesis starts. Okay. Energy is also lost. Remember that line we always say energy is always lost also during respiration, excretion, movement. 90% is wasted. Lastly, as we said earlier about that chicken thing, is that they not many animal parts are not edible. Okay. We're going now to question 74. Explain the role of different bacteria in the nitrogen cycle. Right. So nitrogen cycle is one of those topics you should be revising tonight. What are the role of bacteria? So let's just take a quick look on the nitrogen cycle. What do I see here? First of all, you got those bacteria that live in the root nodules. Those are the ones responsible for nitrogen fixation. You also got decomposers, right? And decomposers do help a lot in the nitrogen cycle because they are gonna break down all the dead matter, right? In addition to this, we have the process of nitrification. Like as you go from ammonium here to nitrites and nitrates, this is called nitrification. And again, bacteria are largely involved in this process. Lastly, it's the opposite of nitrification. What's the opposite word of nitrification? It's denitrification. That is also does involve bacteria. So basically, how many processes we've mentioned here? Fixation, the process of nitrification, decomposes, and denitrification, right? So again, first of all, nitrogen fixing bacteria, the root nodules, they're going to convert nitrogen gas into ammonium. Then we do the nitrification part. They convert ammonium to nitrite and nitrates. Then we got the decomposers that break down proteins into amino acids using enzymes. And lastly, I got the amino acids when they are converted like to ammonium, that also occurs in the soil and that involves bacteria. You may, as I told you, may also write the denitrification part. All right. Specifically here, they're asking me about the importance of nitrifying bacteria. So again, that's the nitrogen cycle once again. It's nitrogen to ammonium. Then you got nitrite, nitrates. This is called nitrification. And then we got the process from nitrates to amino acids. That occurs in plants, right? That's when plants absorb the nitrates from the ground. That's active transport. And again, I'm still in a plant here. That's where plants turn the amino acids into proteins. But like, how are these bacteria here helping? Basically, plants wouldn't be able to get any nitrogen from the environment unless bacteria are there. How did bacteria help us here? They helped us first with the nitrification to convert the ammonium to nitrates. And they also helped us to for the passing on of the nitrates all the way to the plants. Okay. So again, this is absorbed by plants and converted into amino acids. And then where will they be converted? They will be converted into proteins inside the plant. Okay. Okay. All right. Here's like a big question. The question itself may not come as it is, but it's quite useful because it may come in smaller chunks. The question here is asking you to describe what happens to proteins in the human body from the time where you ingest, means from the time when you eat until it's assimilated inside your liver. So again, first of all, proteins are ingested right in the mouth. Proteins are first Like, where do you first ever start to digest protein? You do this inside your stomach by means of pepsin. Then comes the role of the liver in digestion. What's the role of liver in digestion? Your liver does something like this. Your liver secretes bile, and bile is quite important for the digestion of protein, but indirectly. How? Bile neutralizes the stomach acid and allows that pH to be optimum so that the trypsin would work at its optimum pH, right? So bile, which is neutralizes stomach acid to provide the optimum pH for what? For trypsin in the duodenum. Then comes the process of absorption. So I'm going to now absorb those amino acids. Where am I going to take these amino acids? I'm going to take those amino acids like into the blood through the villi. And then they'll be assimilated. They'll be taken from here all the way to the liver. But how? Do you know, like in between your small intestine and your liver, you got a special blood vessel. So this is the guy who ate that protein. This is where we digest it. So here I have like a special blood vessel. I call this the hepatic portal vein. So I'm going to take all these amino acids that I've digested. I'm going to take them to the liver. Over there, we have one of two options. Either you would make use of them, like you turn them to your own protein. That's assimilation. Or second option, you do deamination. You remove the nitrogen part and you convert this into urea. Okay. So again, in the liver, I went through the hepatic portal vein. In the liver, amino acids are going to be assimilated. Proteins. Think of a protein in your body. What could be like a protein you have in your body? Maybe enzymes, antibodies, muscles. So many parts of your body are made of proteins, right? Finally, as I told you, excess amino acids. What are you going to do about those excess amino acids? Those in excess, they're going to be broken down into glucose and urea. And then urea, where is it going to happen to the urea is going to be passed on from the liver all the way to your kidney, right? So this is the route now. We're going to take this to the kidney. And there in the kidney, we will start to remove them, okay? Blood plasma into the kidney where it will be excreted. All right. Right. Another question. Describe how nitrogen protein recycled in the soil into a form that plants absorb and use. Okay. So again, I'm talking here. Let's say we have like some dead animal. How would that dead animal, like the proteins there inside his body, how are these proteins of the nitrogen inside his body, how is this going to get back and recycled into plants? Again, I need bacteria here, right? So first of all, we're going to break down those proteins inside the dead body. How are they going to be broken down? They're going to be broken down by means of enzymes. They're going to be broken down into amino acids, which is converted to ammonium. From here, I will enter the nitrogen cycle again. This ammonium is then going to be nitrified, and like always, nitrates. Plants will absorb what? Plants will absorb those nitrates. How do you absorb ions all the time? Absorption of ions is always by active transport. And it's going to be used to make proteins in the plants. Okay? Next, 78, describe long-term effect of deforestation on the environment. So we're going to talk about this in like three different ways. One about the species, one about the atmosphere, and one about the soil. So I'll divide the answer here into different parts. First, about species. You just say disruption of food chain, extinction of species, reduced biodiversity. And when it comes to atmosphere, you may say less photosynthesis because you cut down the trees. So you expect to have more CO2, global warming, less water vapor. So you would have less rainfall, right? Think about the soil. Cutting down trees causes erosion, floodings. Okay. Think also about the effect on decomposers. Decomposers, we saw today how important they are. So if you cut down the trees, you would also kill the decomposers. But how? Simply because deforestation does what? Deforestation causes the water here in the soil, the water content of the soil to increase. How? The tree is no longer standing here, absorbing the water, right? So for that reason, you would expect to get what we call water logging. And that leads to what? That leads to the suffocation. Those decomposers die because of the lack of oxygen. All right, so let's go back to the question. Here we go. So first of all, I'll say deforestation decreases rainfall, increases CO2. So with the extra CO2 in the atmosphere, you end up having global warming, enhanced greenhouse effect. Also in the soil, you get erosion and flooding. Also, the water content in the soil, water logging, kills the decomposers. And with less decomposers, what's going to happen to the soil content? The soil content is going to be poor. Reason, because we saw how decomposers are important for the fertility of the soil. Also, think about species. Species, you get loss of habitat, disruption of food chain, and that leads to what? That leads to like extinction of species. Here I'm talking about the effect of herbicides. So somebody has sprayed a herbicide. How is this going to affect the ecosystem or the living things in the environment? First of all, a herbicide is meant to kill weeds, right? But indirectly, the farmer is killing also the water plants. The herbicide is going to leak all the way to the water supply. It's going to kill those two water plants here. And with less water plants, you would kill the fish. How? That's not eutrophication by any means. No, you just kill the producers. So all the fish are now less likely to have enough food. And as always, whenever there is any mention of chemicals, herbicides, pesticides, any chemical, plastic, all of these lead to what we call bioaccumulation, meaning the concentration of the chemical will increase along the food chain. Okay? So here we go. Herbicides kill the water plants, aquatic plants, which decreases the amount of food for the fish, the living things in the water. And this decreases the biodiversity. With dead plants, roots, herbicides will lead to what? That would lead to soil erosion. I mean, like we have less plants in the soil, and that would drift the soil away. Finally, herbicides, as I told you, just like any chemical, whenever there's a mention of any chemical, you would mention bioaccumulation. Okay. Number 80 says outline the advantage, disadvantage of pesticides. Okay. First of all, advantage. They are quite helpful. Do you know why? Because when you spray a pesticide, you're improving the quality and the quality of your plants. How? Because you see those pests, they are destroying the plants. But while you're spraying, you're also killing the pollinators like honeybees. And they also leach, they also wash and kill those aquatic animals. Again, you would have bioaccumulation and you'll disrupt the food chains. Okay, so they increase the yield, improve their quality, means the apple would look much better. It's no longer like eaten. Pesticides also kill pollinators. It causes, just like last time, bioaccumulation. You would disrupt that food chain, right? And as always, you would mention disruption of food chain and reduce biodiversity. For 81, I'm talking here, explain the increase of carbon dioxide concentration led to global warming. So again, that's quite important. How does CO2, also methane, how do they both contribute to global warming? Now, both of which are greenhouse gases, right? So what are these greenhouse gases do? Those greenhouse gases are meant, they trap infrared as it's reflected from the surface of our planet. And that's such a good thing, right? That keeps our planet warm, right? But too much of these greenhouse gases lead to what? Lead to what we call the enhanced greenhouse effect, right? So again, radiation from the sun hits the earth, it passes to the carbon dioxide layer around the planet, and here gets reflected back to the surface of earth. This leads to what? This means that the heat is prevented from escaping. Infrared heat is prevented from escaping, right? Causing what? Causing an increase in the temperature of our planet, leading to global warming, okay? Right, somebody wants to check question 80. Here you go. Right. Right. Question 82 talks about plastics. So think about their effect on the ecosystem. First of all, the problem is that they are non-biodegradable, right? There's bacteria can't break down plastics, okay? Second problem is that they are like, they get into the food chain. They suffocate animals, the larger ones. They trap the smaller animals, meaning like if you have a small seahorse in the ocean, that seahorse would get trapped inside a plastic bag or something, right? So, and again, I can mention bioaccumulation here, right? So again, say plastics are non-biodegradable because they cannot be decomposed, broken down by bacteria. They get swallowed, so like a dolphin, get suffocated. It's like a small seahorse, get trapped. Also think about it. If you have a plastic somewhere in a lake or river, or even on land, they'll trap, they prevent the light, right? They prevent the light from getting to the plants. And that kills the plants as well. So they kill plants, they kill animals. Okay. 83 talks about, that's like a generic question. Like I made this question, like I took off a word here and words like example. But and again, every time they ask you about any named chemical whatsoever, what's the route you're going to take here to talk about? You're going to talk basically about bioaccumulation. Again, how does this work? Let's think about it. Usually bioaccumulation is mostly affects the large animals. So if you have a large animal like this fish, or even us at the top of the food chain, we are the most affected. And what does bioaccumulation apply to? It applies to plastics. any industrial waste, pesticides, herbicides, basically any chemical. Why does bioaccumulation happen in the first place? It happens because the small animals, they eat small portions of it. But those small animals live short life. The larger animals live longer and eat more of those tiny plants, right? And usually the larger the animal, the more likely it would live and the more likely it would eat from those tiny, smaller animals. That's why the concentration builds up along the food chain. So one more time, regarding bioaccumulation, and be right here about it. So chemicals get into the food chain. What happens to the concentration? The concentration increases. That's what I call bioaccumulation, causing the death of animals on the top of the food chain. So those are the ones that are mostly affected. And as always, what do I say? I say this causes disruption of food chain, reduced biodiversity, extinction of species. Those are standard words in any of those questions, okay? Okay. Let's just fix something. All right, moving on. Describe impact of large-scale monoculture on the environment. What does monoculture first stands for? It stands like for a farm that looks like this, right? And what effect does this have on the ecosystem? Usually any farming of any type involves what? Involves pesticides, herbicides, right? Involves fertilizers. So just think of any fertilizers and its effect on the environment. I mean eutrophication. Pesticides, herbicides, we've seen their effect already, right? Bioaccumulation, all these stuff. So again, I'll start like this. I'll start with spread of disease. How? That's because you have too many plants of the same species very closely grown together. Also, the farm itself took place, like took over a forest. So you can speak about the effect of deforestation. You may see a decrease in biodiversity, disruption of food chain, all the things I've mentioned, right? Also, it involves pesticides. So I say you kill pollinators. It would also affect like fertilizers, also involve fertilizers. I say eutrophication. Okay. The next question about eutrophication, how are they going to ask you about? Obviously, they're not going to tell you talk about eutrophication. They're going to give you either sewage, animal waste, fertilizers. All three of these lead to eutrophication. Okay. So again, how does this work? It works like this. You've dumped some nitrogen fertilizer or sewage here. First thing that happens is that you get the algae growing. Those algae, they grow on the surface of the lake. They cover up the light, so the light can no longer get to the aquatic plant underneath. And those plants would no longer be able to do photosynthesis, right? Second, plants die, algae die, right? As both die, what's going to happen? You will increase the number of the decomposers. So the decomposers will increase and consume all the oxygen. And that leads to what? That leads to the death of those fish, right? So again, nitrogen compounds are washed into the water supply, causing these algae to grow fast. They block the light. This causes what? This causes the death of the plants underneath, right? And that leads to what? That leads to the decomposers increasing in number. With less plants, there'll be less photosynthesis, less food, less photosynthesis. Also, decomposers would consume all the oxygen. Do you know how they're going to consume it? They're going to consume it by aerobic respiration. Now, I don't have oxygen. I don't have plants anymore. So the fish in the lake will no longer have food, oxygen. Therefore, they will die out. Okay. And question 86 talks about how can governments maintain stocks of fish, meaning like how do we conserve fish stocks? There are many ways you can do this, right? One way is to build fish farms. You could do control the net size, meaning like the size of the net would be big enough so that baby fish will escape. Okay. You could also educate people about the importance of conservation in general. That applies for all conservation, by the way. Like if any question comes about conservation, you may also talk, you can always talk about education. So once again, you could build fish farms, reserves, and applying captive breeding program. What's the difference? Fish farm is like something like this. And within that fish farm, you breed the fish, like you encourage the fish to breed. Also, you can regulate fishing by limiting the time and size of the fish that can be caught. What do I mean by time? I mean like you can only fish at certain seasons. Governments should monitor like the fish stocks and making international laws, like the different countries will make agreements to set how much fish they can catch. These rules must be imposed. What does that mean? It means these rules, you must force people to apply the rules. But how do you like impose a rule? You can put fines and you can also raise the awareness by education. And obviously, you could also make advertisement like public campaigns so that people get aware about the fish. Okay. For 87, it says outline possible benefits of conserving the specific area of a woodland. Again, if you're conserving a forest, how is this going to help the ecosystem? Basically, what am I going to do here? I'm going to reverse the deforestation. Remember when we did deforestation? I could reverse these and get the marks for this question. So again, I'm going to protect the habitats and the nutrient cycle. What do I mean here? Remember when we said that deforestation kills decomposers? If you protect the forest, you are protecting the decomposers and the nitrogen cycle would still be fine. Also, to maintain biodiversity and like disruption of food chain, prevent any disruption of food chain. Conservation of habitat would also prevent extinction. Since plants and animals, they provide what? Like, why should I conserve plants and animals anyway? Why should we be concerned about them? That's because plants and animals, they provide us with what? They provide us with important products. So many of the medicines you buy come from plants. Also, conservation of forests prevents soil erosion and flooding. You may also talk about the global warming. You may also talk about the biodiversity. So everything we talked about there in deforestation, you could just reverse here. For 88, explain the risk to an animal as a result of a decrease in population size. What does that mean? This means you have some animal population, like pandas, for instance. They're just about to get extinct, okay? What effect does a small population have on the species of any endangered species? So again, first of all, it starts like this. You have a small habitat with just few pandas living there. Those pandas find very difficult to breed. Reason, there's just small number of them. So this leads to what? This leads to what we call inbreeding. Okay? First, it's difficult to find a mate. This leads to inbreeding. Inbreeding means members of the same family are breeding, which increases the chance of genetic diseases, meaning if I could like have two pandas here, both are heterozygous. They are healthy, right? But that offspring here will be homozygous recessive, which lead to what? Which causes it to have genetic disease. Also, it decreases in genetic diversity. I'm gonna here reverse that about evolution. That's like de-evolution here, meaning I have less diversity, so less variation, less chance of adaptation and evolution. Also, finally, the most likely effect here is what? The most likely effect is that this would lead to the extinction of the species. Okay? Right. Going now to the next part. Here, it's specifically talking about cheetahs, but again, this applies to any endangered animal. So just why cheetahs are at a risk of becoming endangered, and how can you conserve an animal, right? So again, first one is really easy, right? You can say anything that would kill an animal in its habitat. So it may say something like hunting, disease, destruction of habitat. You can say pollution, global warming, all apply, right? No problem here. Next, describe how species like cheetahs can be conserved. To conserve a species, you could build reserves, right? Remember that fish farm, but we can make it on land. You could do a captive breeding program. You educate people, just like the fish, right? And also, you may also make laws to prevent their hunting. We're getting now to the last 10 questions. Number 90, they say, describe, explain what happens to the number of live bacteria shown the stage labeled A, B, C, and D. So I was given the population growth curve, like the one I have here. And they wanted me to talk about the events of the population growth curve. So again, usually for every stage here, there's one important keyword. Here is adapting, the lag phase, right? So lag when the species are adapting. When there's log, that means they are no limiting factors. As I get to the stationary, that means I start to have limiting factors, right? Meaning I start to run out of glucose, oxygen. During the death phase, right? During the death phase, that's the time where I run out of resources completely and I build up waste materials. Okay, so one more time. Every stage comes with main events. We got lag and still adapting, still making enzymes. We got the log and the fact that there's no limiting factors. We got stationary. During stationary, we are starting to run out of resources. And we got the death phase. That's when I run out of resources and I build up a lot of wastes. Okay? So let's start with lag. They're adapting, right? They're growing slowly. And reason, now I'm going to the log. During B, I mean the log phase here. Bacteria divide rapidly. Sorry, I missed this one here. The bacteria are absorbing resources and making enzymes, right? Again, I'm going now to B, log phase, because there are plenty of resources. Can you name like 2 resources? Could be like glucose, oxygen, any other nutrients. If it's yeast and anaerobic respiration, I don't mention oxygen, okay? Again, during the log phase, the births are way more than the deaths. Stationary, here you start to run out of resources, right? You start to have limiting factors. Finally, the death phase, that's when I ran out of resources and I build up wastes. Good. 91, describe how structure of the heart enables it to pump blood at 2 different pressures. So one more time. You got the heart here. How can the heart pump blood at high pressure from the left side, low pressure from the right side? This has to do with the muscles. This has to do with the muscles in the wall of the artery, meaning the left side here got thick muscles. Here's thinner muscles. Those thick muscles on the left side, they pump blood at high pressure everywhere in the body. The right side pumps blood at low pressure because it's only getting to the lungs. So again, you got ventricles divided by the septum. The right ventricle has thin walls because pump blood as far as the lungs. The left ventricle has like thicker muscles, so it can pump blood through the aorta everywhere in the body. For 92, explain how capillaries are adapted for their function. So again, capillaries, if this is an artery here, right? And this is a vein, right? So how is this, what's the role of capillaries in this thing? Okay. Capillaries come with very thin walls. So the wall of the capillary is quite thin. Thin is good when it comes to diffusion, right? Thin is short diffusion distance. Another thing is it got pores. Those pores are helpful. How are they helpful? They are helpful to allow the diffusion of nutrients, water, right? So again, thin wall is good so you can diffuse oxygen, and the pores are also good so you can exchange materials. Okay? So again, capillaries have thin walls, so short diffusion distance for oxygen, carbon dioxide. Capillaries provide a network to increase the surface area. I mean, that's the network here. You got pores, so substances can move in and out easily, just like what happens in your glomerulus in the kidney, right? Filtration. For 93, explain how a mutation results in an abnormal enzymes does not catalyze a chemical reaction. Okay, so think about it. Mutations happen in the DNA, right? So if the DNA here got an error, this leads to what? This leads to some amino acids being different, because like the order or the identity of amino acids here, that depends on what? That depends on the code you've copied from the DNA, right? Like everything you have on the DNA first gets copied to messenger, and the messenger tells which amino acid is which, right? But what if the DNA has an error? Then you're going to copy the error and you're going to change the amino acid. Okay? So a mutation, I'm defining the mutation first, change in DNA. The change causes a change in the messenger. And this results in a change in the amino acids, right? Here I'm going to talk about enzymes, right? Because the question says an enzyme. Guess what part of the enzyme is going to be different now, which really matters, is going to be the active site, right? So now the active site is changing, right? And when you change the active site, you will no longer be able to bind the substrate, right? So as a result, the protein changes and the active site changing, and now you can no longer bind a substrate. It's no longer complementary. Okay? For number 94, describe how too much fat in the diet may cause coronary heart disease. Fats are what? Fats, they build up like this, right? Fats build up in the wall of the artery, and they limit the amount of blood. So you'd have less blood going to your heart muscle. With less blood getting there, what's going to happen now? You would have less oxygen, glucose get in to the cardiac muscle. Do you see that heart muscle here? Those are the coronaries, and those are blocked. So this part of the heart muscle can no longer get any oxygen, right? That means that this section of the heart muscle will die. So again, it happens slowly, it happens over time. The fat deposits, and this reduces the flow of blood. So you get less oxygen, glucose reach into the cardiac muscle, and less, less respiration, leading to death of that organ. That's a heart attack. Okay? '95 talks about how a molecule of CO2 can diffuse from the air outside all the way to that leaf. So once again, that's the leaf. If you have a molecule of carbon dioxide right here, how is this gonna go into the leaf and get into the chloroplast? Because this is a site of photosynthesis, meaning where photosynthesis occurs. So again, first of all, since it's a gas, I'll just say it diffuses through the stomata into the air spaces until it gets where? Until it gets to the chloroplast. Inside mainly which cells that do most photosynthesis is going to be the palisade. And right there, it would be used for what? It will be used for photosynthesis. '96 talks about xerophytes and their adaptations for survival. Again, what are the limiting factors in a desert? A limiting factor in the desert would be heat, lack of water, right? So xerophytes are adapted to prevent the loss of water. But how's this done? You may have like thick waxy cuticle, no leaves, but spines. You may have hairs so that you can trap humidity. And you would have long roots, so you get water from the source. Finally, you may even have shallow roots, so you can get water as it rains. Like once it rains, the roots will suck all the water from the surface. Okay? So once again, they contain deep, so you can get water from deep underground, or shallow. The leaves got thick waxy layer, and they may also contain fused stomata. Why fused stomata? So the less the stomata, the less the transpiration or loss of water vapor. Leaves are also rolled. What's the benefit of like a roll leaf? If the leaf goes like this means what? This means it's going to trap humidity, right? Xerophytes have like spines, something like this, and toxins. Why these are helpful? those are meant just to prevent being eaten, because they're like very easy, tempting for camels, animals in the desert to feed on. So they protect themselves that way. Okay. Next, we're gonna work on questions about inheritance. So I'm gonna solve like 3 sample questions. First, it's like the normal basic inheritance. They gave me red fruit bred together, and I ended up with yellow fruits. What does that tell you about red and yellow? Since the majority are red, this means it's the dominant. And if you check the ratio is like nearly what? That ratio is nearly like 3-to-1 or 75 to 25%, right? So again, what am I supposed to write here? Letters represented the allele of red fruit. So let's put like R. And here I'm going to put small r. And if you are breeding red and red, I may choose like this, but that would be wrong. Why would that be wrong? Because this will not give you any yellow fruit, right? So what would be the best genotype to write? I'll write it like this. Again, that's the only way you can get that 3-to-1 ratio. And it gives you what? And it gives you the yellow fruit. So again, R, then the Punnett square. Always draw a Punnett square if they ask you, or even they did not ask you, because it gets you all the marks for the question. So that's the Punnett. And again, I already wrote the offspring genotype. I don't need to write them again, but I'm just going to write them here to show you. Finally, write the expected. What does expected means? Like based on the results, it's 3-to-1. So say 75 to 25%. And that's they observed. Research carried out some experiments on tomato, but they got like red tomatoes, but they don't know what their genotype. Just like this. You got some brown rats, but you don't know their genotypes. What would be the way you could identify the genotype of those rats here? You breed them with white rats. White is recessive, okay? So if this rat was R capital, R small, you get 50% white and brown. But if this rat here was R capital, then what are you going to get? You're going to get 100% brown. So again, that's what I call test cross. If it gives you 50-50, that means that the parent is heterozygous. Okay? If it gives you all 100% brown, that means it's homozygous. Okay? Someone's asking, do I write R capital, R small twice, or do I just... No, you'd better write it. Or you may put 2R capital, R small, but it's not going to harm you if you write it twice. No. So again, how do I describe that test cross? I say like, perform a test cross, breed them with homozygous recessive breed. Oops. So I'm going to breed here with homozygous recessive for the genotype. I mean here by what? I mean the yellow, right? Okay. It's not correct to simplify. No, it's correct to simplify 3-to-1. So you may write the ratio as 75-25 or 3-to-1. My other one here is that codominance thing. So what do I have here? I got A, B blood groups, and I want to prove to them that it's possible, if the parents are like this, to have a child whose blood group is O. Okay? So again, you got parents here, and they got children, and one of the children's blood is O. So how is this possible? You're just going to write the parents' gametes here. And that's the other parents. And you breed them together. So that's the first child showing codominance. That's the second child showing blood group O. That's the third child showing blood group A. And that's the child that they asked me about. What's the phenotype of the children? I say blood groups are like what? Are A, B, AB, and the one we're looking for, O. And the probability of an O is 1 out of 4 or 25%. Here it says what? Explain why ABO is an example of codominance. Simply because of what? A and B are equally dominant, right? A and B, none of these is dominant to the other. So they're both expressed at the same time. Okay? Someone is heterozygous, he's not going to be A, he's not going to be B, he's going to be both A and B. Right? 99 is another, that's sex-linked disorder. In my case here, they're talking about color vision, color blindness. We got something like this. That's the genetic diagram. They said, describe evidence that shows that this feature of color vision, how is this showing? How is this, like the diagram tells you that this is sex-linked? Again, let's take a look. Here is the parent. The parent here, there's a father who's colorblind. There's another child who's colorblind. Apparently, only males, the squares, only males are colorblind. There's no single female in the diagram that's colorblind, except this one. Also, only females are carriers, right? Only females show in the carrier. So again, something like this. She could be colorblind. How can she be colorblind? She could be carrier, healthy. The only way she could be colorblind if she's homozygous recessive, right? Like this. But males can be healthy or non-healthy, but never carriers, right? Because they only got one copy of color vision, right? So again, I say because there are more males than females, and only females can be carriers. That's an X chromosome linked, right? Okay. Number 100, the last question. We got color vision, a male with normal, healthy color vision, and a female who's colorblind. First of all, they said write the parental gametes. So I'm just going to copy them here. And next, I'm going to do the usual Punnett square. So what is it like here? It's going to be X. That's the mom. That's the dad here. And that's the mom. All right. And that's heterozygous, right? Or carrier female. And that's another carrier female. And that's a male who's colorblind. And that's another male who's colorblind. Okay. So I already wrote the genotypes. The phenotypes, I'm going to say carrier female. All right, so carrier female twice. And I have 2 colorblind males, right? Okay. They wanted me now to write the ratio. Probability that the baby is born colorblind. What's the ratio I have here in the Punnett square? You can see that only males, half the offspring, are colorblind. Therefore, it's going to be like 50%, right? Okay. All right. We're gonna take 5 minutes break, and then we will do the last part of this session, right? So I'm gonna just pause for 5, and then we will do continue with like extra revision, all right? Right, so we'll continue. First, I'll talk about like description, where they ask you to describe a graph, like what would be the easiest, best way you could describe any graph. There's always a way you could like make your life easy. First of all, let's say this is my graph here, and I was asked to describe it. It looks complicated, but it's always the like easy way you can get marks out of it. So usually I start like this. I say like as some factor increases, another factor increase or decrease. In my case here, you can see that with time, the enzyme activity is increasing for both of which, right? All the way like to day 11, right? So in both cases, there's an increase, right? Also, I may say like as x increases, the y increase or decrease, okay? So again, start by describing the entire curve in one sentence. Overall, there's an increase, decrease. And then specifically say as this factor on x, like time, the other factor on y, enzyme activity, changes. Then I say it peaks at specific dates, like day 11 here, for instance, right? And it levels off. What does level off? Means like my graph, for some reason, went like this. The graph is leveling off. It may increase, decrease rapidly. Like, for instance, here in my case, there was like a rapid, slow increase, rapid increase, okay? You may also say gradually or slowly rather than rapidly. And when you see the curve does like this, like the one I have here, you say, well, the change is, or the graph or the data is fluctuating, right? Or you may say, if you don't like the word fluctuate, say, well, there is no significant change or increase or decrease. Okay? Next, I want just to help you with the keywords for tomorrow. So I'll try to remind you of the keywords related to each of our topics. So in the lesson of classification, there is morphology, anatomy, and the fact that using DNA is more accurate. So again, the old system was based on morphology, anatomy, and the new system is based on DNA, which is more accurate. In the topic of membrane transport, active transports, when do you write about active transport? Whenever there's a mention of ions, and it's always against the concentration using carrier protein. Whenever there's a mention of water is osmosis, and here you mention what? You mention a partially permeable membrane. ppm itself is not acceptable. You just write it like fully, partially permeable membrane. Again, those words in black are like the package. If I write about osmosis, I write about the partially permeable membrane. If I am talking about active transport, I talk about respiration. If I'm talking about diffusion, I mention the concentration gradient. Okay? In the lesson of enzymes, there are many keywords here. You may mention substrate. You may mention the enzyme substrate complex, the active site, the fact that enzymes denature, and the fact that enzymes work at an optimum pH. So again, when do I write the word enzymes in general? Anywhere where there is one substance turn into another, like protein to amino acid, I say enzymes. Nitrates turn into, ammonium turn into nitrate, I say enzyme. Any mention of a chemical reaction, say by means of an enzyme. Okay? If there's any mention of magnesium nitrates, with magnesium, you got chlorophyll, photosynthesis. With nitrates in plants, you mention growth and amino acids, proteins, even DNA applies here, okay? For photosynthesis, there's always that question about limiting factors. And usually, you really need to take a look at the x-axis. if you got light intensity on the x-axis, this means what? This means if the graph looks like this, the limiting factor here at the beginning is going to be light. And the limiting factor here, as the line becomes leveling off or steady, is going to be either CO2 or temperature. Right. Next, wilting. Some question about wilting. When does it happen? Wilting occurs when there is more heat and less humidity. Right. So again, factors that would lead to wilting. That means there is more transpiration, meaning more heat, less humidity, more wind. On the other hand, the plant is getting less water, meaning the plant is absorbing less water from the ground. What does that lead to? It leads to less turgor pressure, and the plant cells turning flaccid. Okay? My last one here is translocation. When will the leaves be a source? Leaves are the source when they're present or when the light is there. And when will the roots or storage organs be the source? Simply when there is no light or no leaves. Okay? Next, if there was any mention of transpiration, again, like I'm going to track the flow of water inside the plant. First of all, it starts with root hair cells. Then it moves to the cortex cells. I'm still in the roots here, right? Then we got cohesion in xylem. Then when we get to the leaf, you got evaporation. Where does it happen? It happens on the spongy mesophyll. And diffusion of water vapor through stomata. Okay? Regarding bile, bile is emulsification, right? That's when we turn fats into small droplets. That means what? That means you got more surface area, and lipase can now break down food faster. But that's not the only job of bile, because bile does what? Bile is also to neutralize stomach acid, so that what? So that the enzymes of the pancreas will work on their optimum pH. Those questions that involve the whisking tube, looks like this. That's the whisking tube here, right? Whisking tube, to start with, is partially permeable, means it has holes in it like this. And again, small particles inside the whisking tube would escape, right? Larger ones like starch would not escape. But if you digest them, when they get digested, they'll be able to escape. Cholera, first of all, it's a bacterium, right? Meaning that it's going to be killed by antibiotics. In addition, you would treat it with rehydration, meaning you give the patient water and salt and sugar. How does it cause the problem? How does it cause people to have severe dehydration? Simply because cholera secretes a toxin. The toxin makes you or makes the patient lose chloride. And with more chloride, this means what? This means that the patient will lose a lot of water. Here I'm talking about the gas exchange surfaces, like the ones you have in your alveoli, or the ones you have in gills of fish, for instance. First of all, the walls are thin, right? Second, there's a lot of surface area by means of the alveoli or the capillaries. Plus the action of breathing. When you breathe, when your blood is flowing, that means what? That means that you are having a steep concentration gradient. Okay? Also about gas exchange. The short diffusion distance. How? Because of the thin walls. The large surface area, because you got so many alveoli and so many capillaries. I think I'm repeating myself. Sorry. one more now. It's the effect of exercise. I mean, the guy here is working out during the exercise I'm talking here. So during the exercise, the athlete or whoever's doing the exercise will have to work out his muscles. And you know, like when the muscles contract faster, that means he would require more energy, so more respiration. Okay? His breathing rate and heart rate will increase, right? So that what? So that he would supply more oxygen, glucose. He's trying his best to do what? He's trying his best to maintain aerobic, and he's trying his best at the same time to prevent anaerobic. So again, this was during exercise, okay? What about after exercise? Here you deal with the oxygen debt. So again, this is after the exercise. Now, in the muscles, there wasn't enough oxygen. The athlete is not trained enough to supply enough oxygen to his muscles. So lactic acid starts to build up. It diffuses out of his muscles into the blood, and then it goes to the liver. Once in the liver, what's going to happen? He's going to break down the lactic acid aerobically to produce glucose, carbon dioxide, and water. Okay? All right. Next, it's the kidney. You got two processes in the kidneys. One is filtration. That's what you do in the glomerulus. So here is the fact that small and soluble materials like glucose, urea, salts are forced out, but not the large particles like proteins or blood cells. So the filtrate will escape out, and then we will reabsorb. You'll reabsorb the ions and glucose by active transport, and you will leave behind urea, salt, and water. Okay? Reflex. You have two reflexes in your eyes. You have the one related to distance and the one related to like brightness. So let's start with the brightness first. If it's too bright, then your circular muscles will contract. That reduces the size of your pupil. And if you're looking at something close, here you're going to work out your ciliary muscles. The ligaments, meanwhile, they'll slacken, means they become loosened, and your lens is going to get thicker. Homeostasis, if it gets hot, then you do vasodilation of your arteriole. You start sweating, not sweating, sweating, sorry. You know the spelling, right? Sweat. It's actually correct, sorry. Too cold, what's going to happen? If it's too cold, you would vasoconstriction. I'm talking arterioles here. Hair stand. And you may also start shivering. Last bit of those keywords is the fermenter. How do you maintain optimum temperature and pH in the fermenter? You would use like the probes and the water jacket for temperature. And how do you supply nutrients? You supply the glucose for respiration. Don't forget that you would also need to supply a source of nitrogen, like amino acids. How do you prevent contamination? You prevent contamination by means of steam or bleaching, so that you would maintain the conditions so you don't grow pathogens, basically. Restriction enzymes. I'm talking here genetic engineering. You got two sorts of enzymes. The one for cutting DNA, restriction. And when you cut DNA with restriction, you will make those complementary or the sticky DNA ends. But you also need to combine pieces of DNA. The one ligase. Ligase like fuses DNA from bacteria, plasmid, and humans. And you make that hybrid plasmid, half human, half bacteria, recombinant. What are the advantages of self-pollination? It's guarantee, right? Ensures pollination. It saves energy because you're not like dumping pollen grains everywhere. You don't rely on pollinators. And if the parent is successful, if the parent is well adapted, then the offspring will inherit all these good features. Okay? What are the disadvantages then? Disadvantages are all related to lack of variation, overcrowding, less adaptation. Again, I'm going to talk here about the events that lead to extinction. So I'm ending in extinction. So less adaptation, less survival, and less chance of evolution. The menstrual cycle, you got two sorts of hormones. You got the LH. LH is the one, yeah, there is a chance of genetic diseases too. LH is the one responsible for ovulation. And LH is the one that stimulates progesterone. Progesterone is the one to maintain the lining of the uterus and is the one that inhibits FSH and LH. So again, I need to go in order here. So we start with FSH. FSH is the one to start the cycle because it matures an egg cell, stimulating estrogen. Estrogen is the one to repair, and it's the one to stimulate the next hormone. What's the next hormone? It's LH, right? So I'm going to stimulate LH and block FSH. All right. So I'm reaching now to calculations. Easy calculations you get. One, it's percentage increase. How do you do percentage increase? Let's say I was given this graph and was asked to do percentage increase. You find the difference. Here they wanted me to find the difference between 1940 here and 2020, which is right here. So I read from the graph at 1940, it's like here it's 20. And here it's like 136. So first of all, I'll find the difference between the numbers. And then I'll divide by the lower value, the one I started with. How much did I start with? Started with 20 times 100. Okay. But what if it was percentage decrease? Let's say I was given these numbers and I was asked for percentage decrease. What are you meant to do here? You're meant to find the difference once again. So I'll find the difference between the numbers. And I'll divide by the starting value. But in this case, your starting value is the larger number, which is this one, 79%. Okay? You would only put negative positive signs if the difference gives you negative positive. If the difference gives you a positive sign, you keep it positive. When you get positive negative signs, you would get it usually either if you have percentage decrease or if you have percentage change. Percentage change is very similar. You only find the difference divided by the starting value. Okay? That's percentage change. Okay? I also want to get like quick recap on most important, like 10, if you are to study 10 questions, what are these 10 questions that make big difference tonight? First one is the one about the food chains, the one we solved today, right? So I'm just going to remind you of the keywords. First, you say energy is lost by Mrs. Grant. Second, you say a lot of animal parts are not edible. Third, you say 90% is wasted. Then you say little energy is reaching the top animal, carnivore. Finally, you say there's not energy enough to support that top population. Remember, we solved today that question? So just quick reminder. Okay. Next, it's the effect of a chemical. If they mentioned any chemical, regardless, how do you speak about this? Let's say this guy is spraying a chemical in the field. So what effect this chemical will have on that food chain? The key word here is bioaccumulation. So you say it leads to bioaccumulation. First, you say it enters the food chain. The chemical builds up, means it increases along the food chain. The top carnivores are the ones mostly harmed. And as always, I say decreases biodiversity, disrupts food chains. Vaccination and active immunity. So again, somebody got vaccinated, right? And how does that lead to active immunity? You start like this. You define what a vaccine is. You tell them it's weak or killed pathogens, stimulates lymphocytes. When the lymphocytes are stimulated, what are they going to make? They're going to produce antibodies, and they're going to produce memory cells. So again, they're going to make antibodies and memory cells. And that provides what? That provides long-term active immunity. Sewage, fertilizers, and eutrophication. Again, we did this one today. So just a quick reminder, you start with the algae. Then the algae block the light. You kill, the algae die themselves, and the plants die. And that leads to what? That kills the fish in the lake. So one more time, the algae increase in number. The lack of light. The aerobic respiration increases. That means the decomposers are consuming the oxygen here. The lack of oxygen kills the fish in the lake. Monoculture farming. Again, we did this today. You're growing a farm of one species. Again, the key word here is that diseases will occur. So you have one species. You define for them. Again, if there's a word that is not familiar, the best way to start is define the words. Like in case of vaccine, I define the vaccine. Monoculture, I define what monoculture is. Then I say diseases spread, less biodiversity, disruption of food chain, and talk about pesticides, herbicides, okay? Okay. You want the question before vaccines. Is this the one you want, Reem? All right. Next, it's this one. It's talking about natural selection. So here I'm talking about variation. Again, how's the question going to come? They tell you, well, like, how did giraffes get the long neck? How did the animal become better adapted to its environment? So I start like this. I say variation due to mutation. I say that the less adapted animal dies out. The more adapted one survives, reproduces. So again, mutation, a new allele. You have to use the word allele in this context. The adapted animals survive and reproduce. The allele passes to the offspring, and that's called evolution by natural selection. 4 questions to go. Then this question here says, explain how an impulse transported across the synapse. Someone's asking you, how does an impulse reach from the presynaptic to the postsynaptic neuron? So how does this start? It starts with the neurotransmitter diffusing from high concentration here across the synapse, and then it binds to a receptor in the postsynaptic neuron. So again, you have vesicles. Do you see those vesicles? Those vesicles move towards the surface. They release the neurotransmitter, and then it binds to receptors in the postsynaptic neuron. And then you form like start a new impulse in the postsynaptic neuron. Someone's asking, what's the difference between percentage change and percentage decrease? The same. Every time someone asks you for percentage change, that is what? That's difference over the starting value. So you divide by the starting value times 100. What is percentage increase? Again, you would divide by the starting value, but your starting value is going to be the lower. So this could be a low value, that's percentage increase, or could be the higher value, and that would be percentage decrease. Okay, so they're all the same thing. You're always dividing by the starting. It just happens if it's percentage increase, you divide it by the lower, and if it's the decrease, you divide by the higher. Okay. There's no marks deducted over spelling, okay? Yeah, you divide by the old one always. Next, explain why athletes continues to breathe after exercise. So somebody's doing an exercise, but he continued to breathe fast after they exercise. How is this gonna, why is this happening in the first place? That's because he made lactic acid, right? Inside his muscles. That lactic acid moves from the muscle to the liver. Over there, he's going to break down the lactic acid by aerobic respiration. So here, anaerobic respiration took place in his muscle. The lactic acid built up inside the muscle. The lactic acid is transported in the blood plasma, like to the liver. And right there in the liver, he's going to break down the lactic acid aerobically. So here is aerobic respiration, and the muscle is anaerobic, okay? What are the stages that lead to cholera? It starts with a bacterium. That bacterium releases a toxin. The toxin affects the epithelial cells of the intestine. And those causes what? That causes the loss of chloride. The chloride ions lead to what? The chloride ions drop the water potential. And as the water potential is decreasing in his intestine, that would lead to what? That would lead to the loss of water. So again, toxin and release in the small intestine, loss of chloride, lowers the water potential, and the water is lost by osmosis. That's the dehydration part. Very last question. Explain how insulin controls the blood sugar. So insulin is made in the pancreas. It travels to the liver. So insulin is when the blood sugar is high, she's eating. So her blood sugar is going to increase. That's where insulin is secreted. She's eating, see? Her blood sugar is increasing. That leads to what? That means that sugar, glucose, is absorbed into the blood and is converted. That glucose here is converted to glycogen. If she's working out or she's fasting, starving, then it's going to be glucagon, right? All right. Again, I start like this. Pancreas secretes insulin. Liver absorbs glucose. Liver cells convert glucose to glycogen. And as always, since I said the word convert, I mentioned enzymes, right? Enzyme worth 1 mark. And I restore homeostasis. Okay? Right, so we will stop right here. I'll wait for a bit here to see if you have questions. If you don't have questions, then I'll let you go and revise. So percentage decrease, okay? So you started with 50 and the value dropped to 25, right? So how much is the difference in this case? The difference is 50 minus 25 divided by 50 times 100. So it's positive 50%. Here is negative. I agree. So here is going to be negative, right? So it's going to be the final and minus the initial. So that's the negative, right? But if it's the other way around, it's going to be positive, right?